So what I'm ambitioning to do in lecture today is not necessarily finish every one of the groups, because that hasn't typically happened, but at least getting through the topics on complement. Now, why I'd like to do that is because that will leave you with all the information you will need to start writing essay question 1. And this question is, you'll see, jumps in without really covering some of the general information I gave, comparing and contrasting innate and adaptive immune responses. So clearly that information is kind of scattered or irrelevant to multiple questions, to all 3 of the immune questions. And that was more to give you a frame of reference as we go through the details of this. So the essays are focused more on those details. So the first part of the essay asks you to compare macrophages, which the only ones I picked out were the free macrophages, just to make the fixed ones are largely dendritic cells, it's believed, not totally clear, but largely dendritic cells. So not to worry about the fixed versus free. I would know that definition because you're comparing the free ones to neutrophils and dendritic cells. And since many dendritic cells, although probably not all of them, are fixed macrophages, then it's just what are those features. So we're going to have the group that was group, if I remember rightly, group B is doing that, part of that. They're doing the neutrophils and macrophages. They're talking about similarities and differences for, that's lecture question 5. But we'll spend some time extending that and talking about dendritic cells again. And dendritic cells will be one of our first entries into, it's part of the innate, but it plays a critical role in the adaptive. So that's the plan. And to help you with this is actually what I would have posted for helping you with the, with writing this first essay is a table that I generated a while ago. And back then, the first essay also discussed natural killer cells. That is now moved into the second essay. So you'll see this table and we'll fill part of it out. But what I gave you is a blank one because what I'm writing here is not all or only thing you could say. So it'll depend on how easy it is for me to write much into the table as we go through the group B's answer and then extend it a little bit. Okay, so we now still don't have everybody here, but I'm going to go ahead and break you into groups, and then people who come late, we're just going to be stuck randomly. So if you...
Why are we doing it?
Somebody have their mic on inadvertently, or do you have a question? Looks like I got to turn the microphones off.
Professor, I have a question.
Prabreed, is that you?
Yeah, Prabdeep. I just wanted to make sure. We haven't moved on to lecture question 6 yet, right? We're still in lecture question number 5.
That's right. That's where we're going to start. So if you're in group B, then you don't have to worry about going back over lecture question 4, which was part of your group. You can jump right into the lecture question 5.
Okay, thank you.
Was there another question that somebody was talking about?
I did a little bit. Actually, I did one half of it. I did one half of it in my room.
Yeah. Okay, looks like I just need to turn everybody's microphones off. All right, you can turn it on again if you have a critical question. And I see a few more people have arrived. So So let me just make sure I take note of where you were before. So when I put you in your groups, you'll end up in those groups. Okay. All right, so here we go. I am going to make the breakout rooms for 15 minutes again. And there is one change I would like to make from the posted questions. If I give you group 7, can you do lecture question 13 as well? So the reason I'm doing that is I think 11 and 12 has got a lot of information, and 14 and 15, which is what you do as group F, is all done for you in the table. It's just you looking it up. So I just added 13 to make it a little more balanced. So I will, I'll write that in the name of the breakout rooms too, so you'll remember. Okay, so let me make, how many do I want? 1, 2, 3, 4, 5 breakout rooms. Okay, and I want to assign them manually, create, and we're going to call this one group B. We're going to call this one afterwards, so I don't have a total. But so that group that started this question about similarities and differences between neutrophils and macrophages, that is group B. What I did while you guys were in the breakout rooms is I started to put down onto the table some of the things that you already covered last week or last Tuesday. So let me share the screen that has that table. Now, if you came in late, this table was, and I probably forgot to tell all of you, I put it into files, and under files, you'll want to go into the lecture exam 2 folder and then into the immune folder, and you should see a table that looks something like this one. So you'll notice, as I mentioned to you before we went in the breakout rooms, NK cells are on there. They're clearly not part of SA1 now, they're in SA2. But I left it there because you are going to need to know about NK cells. So when we get to talking about them, which is the very last question in this set of lecture questions refers to NK cells, maybe we'll call this table up again and put things under there. The essay focuses on these 3 cells, and in particular for the macrophages, it's asking about the free types. So that's all I'm going to be talking about here, because most fixed macrophages are probably dendritic cells. So this is what you guys did last time. Let's see if between question 5 and question 6 we can add to this column, and then we will come back because some of the things are not in these lecture questions, but are certainly in the material you need to know, and take a look at some of the other things like cytokines that it releases and what it enhances and its activity. Later, some of the people will be looking at general phagocytotic processes, but part of lecture question 6 does talk about parts of it. And then we'll summarize some of the things at the bottom. So I think this part is pretty much done, right? Is there anything you guys, do you have a spokesperson? And is there anything your spokesperson wants to add to where you find these cells? Who's the spokesperson? Is it Jeanette? Because you're lit up, but I think you.
Yeah, I was speaking last, but we didn't really go in depth because we thought we already covered this last Tuesday. But what you have is pretty sufficient. We didn't really discuss locations of the cells.
So the only thing I would add, and this is coming out of the lab stuff, and you are going to be drawing these slides today and needing to know some of the cells that are found in these various structures, is because macrophages can become antigen-presenting cells, which we'll talk about more later, you do find macrophages in lymphatic tissues. Now, they probably migrated there after they encountered their PRR-PAMP relationship was established, so after some sort of infection. But you always have microorganisms all over you, so you are pretty much always going to have some sort of movement between the activated free macrophages moving back into lymphatic tissues. Okay, so we did talk about what activates them, and in your compare-contrast, the idea that cytokines can activate neutrophils. Now, the cytokines that I brought up are the prostaglandins released by damaged tissues. So we talked about this a bit last time, that in the plasma membrane of all cells, any cell, including the white blood cells, there are echinocanoids, which are little fatty acids that are free in the lipid bilayer. And when those are released, they will cause inflammation, but they'll also cause activation of the neutrophils, as well as calling the neutrophils in out of the blood. As we talk about inflammation later today, you'll see that one of the things you need is a gradient so that the white blood cells know where to go. And the damaged tissue cells are releasing these prostaglandin echinocanoids as a signaling molecule, and that is a cytokine. So be careful. I'm going to italicize this because if I do give you an example, this may not be the only cytokine that would do this, but we do have an example here, then you need to know that example specifically. And one of the things with the book is it often just falls into, cytokine, and then it'll have tables that talk about cytokines that do these things. So I'm asking you to know examples if they're given in our lecture or in the book. So the book does discuss this, and so for your activating of neutrophils, what you have are at least the prostaglandins released by the damaged tissue cells, so don't reply cytokines. Cytokines is such a huge category of items, and we're only looking at a very small number of them, although it doesn't seem like that if you look at the table, but we are, and therefore we're going to have at least one example when you have some specific trigger like this, and so you won't need to use cytokine as a word very much. Is that clear? Anyone question about that? So where will you find it? Well, I made that fuse table, that big table that I think the later group, like group F or something, is going to use, called the innate immunity cytokines and antimicrobial. All right, so we didn't discuss much about what enhances the activity in any kind of specific way, but I did mention last time that your adaptive immune response does enhance it. So I'm going to add interleukins, and this is the way interleukins are abbreviated both in the book and in how I'll do it. And we're not going to give specific interleukins. So this is one place I'm making it a little bit easier. In the book, there is a table of these interleukins, but I am not asking you to know the specific interleukin 6 versus interleukin 10 versus, not asking you to know that. First of all, it's hard to remember numbers later, but generally interleukins, and in particularly, these are interleukins that can be from, let's just come back to this, but from the adaptive immune response definitely can enhance both of these. And for that matter, even the dendritic cells are going to be enhanced. Now, I skipped this because we didn't cover that in the short answer. I should come back. And basically, these are going to be the same as this one. So that's not in the lecture question, but we'll try and go along just for your essays as you work on your essays. So what I'd suggest in this essay is, here's your table, you've got it. You could highlight similarities versus differences, or you could rewrite it. Personally, I think rewriting is a good thing. So what are cytokines that it releases? So here I'm going to recommend you go into that table. I mentioned leukotrienes, and these cause inflammation. They also act as a chemotactic gradient for the white blood cells leaving the blood and entering into those tissues. But there are more. And so I refer you to that table and suggest that you guys fill this out. One of the things I want to stress in general, without filling out what other cytokines, is in general, these guys act more as a general in the militarized analogy, in that they coordinate aspects of the innate. So for example, they're going to talk to the liver as we go forward. We'll see this, that macrophages, once they get into the tissue and they're activated, are releasing quite a few cytokines, some of which are in that table, that then tell the liver to make more of lectins that we'll talk about later. Or they could be releasing tumor necrosis factor alpha that's in the table that may act, you can tell from its name that it tends to cause a degradation of tumor tissues. So it does more than just act locally, it acts at a distance. And therefore, that's how I got this idea of it being the general. It's running the show in the innate response, more than, say, the neutrophils. These guys do release cytokines, but their main point is that they are going to be releasing antimicrobial and being phagocytes, and they are like foot soldiers. So maybe I'll just because it's in the same place, we'll say these are more like the foot soldiers, in that they are going to die in large numbers in the battle of at the site of entry, of the portal of entry. So if you see, say, you have a wound and you see it's a little bit infected, and what you're seeing come out is pus, that sounds great. A lot of what's in that pus are dead neutrophils. There's just so many neutrophils, and they swarm into the area. And I forgot to put the video, but it has a video showing them do this in live animal. So I'll put that video up during your lab time, or we could watch it together during lab. And they zoom in there, but they release these hydrolytic enzymes, and those enzymes do kill them, not just neutrophils, but they kill body cells and they'll kill other immune cells. So, these are dangerous little critters in a way, because they are so destructive. And then they release some specific stuff for antimicrobial, and they do phagocytosis as well. So they're a much smaller cell, if you remember, so they're not able to consume as large entities, and they also don't consume as many entities as the macrophages. So as you're comparing and contrasting, I'm not writing that, but hopefully you're taking some notes. So given that sort of more bigger picture portrait, if we go back down here, then hopefully it's clear that these should be a short-term response, because they're so destructive. You don't want this to go on or be inappropriately triggered, because they are destructive to your body cells. Whereas these guys tend to be long-lived response. So once you get monocytes entering into that area, that oven where the pathogen is located, if they aren't killed by the neutrophil, ones that remain will stay in high numbers in that tissue for weeks afterwards. We'll just put remain for weeks afterwards in higher numbers. And I don't mean in the blood, I mean higher numbers in the like portal of entry where the infection began. And I was distracting myself here because one of the things that besides the general innate response, I'm going to say including the CSFs, because they also help coordinate the bone marrow's response to, and that's going to be via interleukins typically. So they do release interleukins themselves that go to the bone marrow and tell them make more monocytes or make more neutrophils and help coordinate that kind of response. So how about this phagocytic processes? I know you had a bunch of terms to define, Jeanette. Can you help us with some of those terms? And I'm just going to put the terms in here as you go, because
yeah, so this is our next question. So are we doing like phagosome optimization questions on number
yeah,
two?
exactly.
Okay. So optimization, we said that it coats the antigen with antibodies.
You know what? You're a little far from your microphone, so I think it's hard to hear you.
Okay. Can you hear me better?
Yes, that's better.
Okay. We said opsonization, you coat the antigen with antibodies to enhance phagocytosis.
Coat pathogen with, and we'll call them opsins because there's more than just antibodies, opsins. And why do you do that? To, what did you say?
To enhance phagocytosis.
And I'm going to add enhance / permit phagocytosis. So what happens in our war with microbes is that microbes have PAMPs on the outside, and they can't help but have those things because they're mostly structural for them, and so it's very difficult for them to evolve into not having those PAMPs. And so they coat the PAMPs, they hide them. And you'll learn more about this in microbe, but both some bacteria and most fungi will secrete something called a capsule. And that capsule will coat the chitin, will coat the peptidoglycan, and therefore hide it from a phagocyte. Now, what phagocytes are we talking about? Well, we're talking about any phagocytic cell. So I could copy all of this and put it into here, and I could put it into here too. They all are going to respond or potentially require opsonization. So because microbes have evolved a way of hiding PAMPs, then our immune system tries to one-up that by finding a way of getting around the lack of an exposed PAMP. So what you'll learn is, as Jeanette told you, when we talk about antibodies later, they have sites that macrophages can bind to. So if they see an antibody bound to its antigen, then they can grab onto that without using their PAMP or PRR, and they can do phagocytosis. In addition, if there is a coating on it, so a capsule or something, then not all PAMPs are covered. It does, as she say, enhance it. So it makes it a lot easier, gives the macrophage, the neutrophil, or the dendritic cell more handles with which to bind, because you cannot do phagocytosis without binding. So I'm going to kind of put that rule up here. Must bind surface molecule to phagocytize. Oops. To start. I can't spell because it's shifting, as you can see. Phagocytosis. And opsonization can help you. Now that again is true for all of these. So I'm only writing it under neutrophils, but whatever I'm writing is also true for these guys. So once you do the phagocytosis, can you tell us about a phagosome? This is the next word.
So for the phagosome, we said it's basically, it becomes a phagocytic vesicle once it engulfs the pathogen. So after binding the surface molecule of the pathogen, it will create a vesicle, and that's considered the phagosome. Once it's inside the cell, it will fuse with the lysosome and become a phagolysosome, and this is where the lysosomal enzymes will digest the particles. And then from there, I guess oxidizing agents such as the respiratory burst will kill the pathogen. We didn't really go in depth in this, but we just said the peroxisomes within the cells contain enzymes that will oxidize certain molecules.
Okay, great. So lysosome, what do lysosomes have inside them? You may have said it.
The lysosomal enzymes.
I'm sorry, your paper shifted. What was that?
The lysosomal enzymes will digest the particles.
They're hydrolytic enzymes, so they're proteases and lipases and things. Hydrolytic enzymes. Or a peroxisome. And what does a peroxisome have in it?
The peroxisome contains enzymes that oxidize certain molecules.
They're actually not enzymes. They actually contain hydrogen peroxide, so H2O2. And these are oxidizing agents. So now I'm putting it over here, but actually let me take that out of there if you're copying it, because it's these guys that have the peroxisomes. And no peroxisomes over here, as far as I know. So what happens when they release these hydrogen peroxide oxidizing agents? What do you call that?
It's called a respiratory burst.
Good. Okay, so this is just another way of killing. So if you can't digest, maybe there's no particular substrate on that pathogen that the enzymes can act on, well, we have another alternative. This is one of the themes in your immune system. It's complicated in part because it's redundant. And that's because we've, of course, evolved alongside these pathogens who want to use us for food, and we don't want them to use us for food. So, so you end up with different ways of killing. Now, one of the heads up I want to make sure you maybe make flashcard or something about this is lysosome and lysozyme sound very similar. Lysosomes have a whole cocktail of different enzymes in them that digest proteins and lipids are probably the main ones, but even maybe carbohydrates, I don't know. Why proteins and lipids? Because they make up plasma membranes and cell walls, and maybe carbohydrates that make up the capsules would have to be digested as well. So lysosomes have this whole cocktail, and they are there are many different enzymes. Lysozyme in particular digests the peptidoglycans. So they are a specific enzyme, not a whole bag of different enzymes, but their names are so similar that I see students year after year confusing those two terms. So please keep those straight. So I don't know of any peroxisome and respiratory burst in these guys, but it's possible it's true. I think our book just talks about these two doing a respiratory. Well, they only talk about this one, but my guess is that one. They're very similar cells. Okay, so that finishes up your group. Now, what we haven't done here is talk more about how phagocytosis happens, and that's a later question. But before we leave this table, this is more useful for lab because you will need to know what is, and maybe it's more obvious from what you've done with the differential white blood cell count, and you might be able to answer this. But what type of pathogen it works against best would be extracellular pathogens, because what you're doing is digesting things using enzymes, defensins, and particularly for neutrophils, because they are a short-term response, we're going to call this acute bacterial diseases is what the textbook. That doesn't mean to say that fungal diseases, I mean, if you have athlete's foot or something, it's not going to act. But that's where you see the rise in the differential white blood cell. So I'm going to just put increased differential white blood cell count. If you see a rise in neutrophils above their normal level, which is pretty high already, then you suspect in that patient there may be some kind of acute, especially acute bacterial infections. And that's correlative. So it's not they can't do other things. They can certainly phagocytize viruses. And if they digest them, they could phagocytize a fungus. But bacteria is where you see that differential white blood cell. So given the long-lived nature of this, again, for our differential white blood cell, we'll say any, but rise in differential white blood cell count with chronic infections. So the book gives examples like tuberculosis as an example of a chronic infection, which you have at a low level. So do people get the terms? If you don't understand the term chronic acute, can you put that in chat, and I'll go more into what those two situations are. 6 new messages. Let me see what's down here. Okay, nothing new. All right, looks like you got it. Now, I didn't fill this out for dendritic cells because most of the discussion in the book is around its role in triggering the adaptive. So we'll put any here. Any type of pathogen, because if you're going to trigger an adaptive response, then they have to be able to act against any type. And these are certainly going to be longer lived because the adaptive remains high. These are lymphocytes. So for your differential white blood cell count, you would see these rise in number when you have a more longer term infection with an adaptive response, because that's where their main role is going to be. So this is a lymphocyte, and in our differential white blood cell count, we don't expect you to be able to know who's a dendritic cell, who's a B cell, who's a T cell. So because you're just looking in the blood. But these guys, if they're a fixed phagocyte, they're going to phagocytize their pathogen, and then they're going to go back into the blood to get to the lymphatic system so that they can trigger the adaptive response. So what about these other blanks? Again, not clear if leukotrienes, but I wouldn't be surprised. I'll just put ditto, maybe, or everything to the left. But they are certainly sensitive to interleukins, and we'll talk more about that when we get to the adaptive response. And there's no discussion about any kind of antimicrobial. I wouldn't be surprised if they release defensins and lysozymes. They are phagocytes, though, because that's going to be the main job, and they're going to do that to become an antigen-presenting cell. So we will discuss more of these later. So if you write this essay this weekend, you may want to enhance dendritic cells as we go forward. So one other thing we're going to be talking about is the response of these cells to inflammation. So the next group is going to discuss inflammation, and I believe the one after that is going to talk about, how these cells leave the blood and then start their phagocytic processes. So whatever we say about these effects will affect all of them unless they're already there, a fixed phagocyte. So if it's a dendritic cell that's already in the tissue, it doesn't need to leave the blood. So, okay, let me stop sharing this screen. Is everybody good on this? Anybody want another minute on this or some particular thing? Okay, I am not going to share the filled out one because I think it's important for you guys to take notes and for you to fill it out. So if you haven't had a chance to keep up with my typing, then you can ask about it. I'll save this. In fact, and then you can ask me about it, but later in an office hour or in lab. But I'd like you to work on it yourself because I think that's a better way of learning it. Okay, let me stop here and let's go to the next group. And I'll help by hopefully taking some notes here. I've got to turn this on. Somebody's got their microphone on, embarrassingly enough, since that was a toilet, I think, flushing. You might want to turn your microphone off. Okay, so what about inflammation? Who is the spokesperson for Group C?
That's me.
Okay, Dina. So for Essay 7, can you describe how inflammation could be triggered? And there's a lot of answers to this, so.
Well, for question 7, I have describe how inflammation is triggered before white blood cells or plasma components arrive. And I've got inflammation begins with a chemical alarm signaled when damaged or stressed cells release inflammatory chemicals into the extracellular fluid. Prostaglandins are fatty acid molecules produced from the arachidonic acid found in the membranes of all cells. They are released when a cell is injured or stressed. Prostaglandins increase inflammation, trigger the pain response, and strongly promote the chemotaxis of neutrophils. Present in body secretions as well as neutrophils and macrophages, defensins and lysozymes begin to act against cellular pathogens immediately by disrupting and damaging their cell membranes and inhibiting bacterial and fungal growth. Mast cells and basophils present in the connective tissues release histamine in their granules, further promoting inflammation in response to a mechanical injury. The presence of certain microorganisms and chemicals released by neutrophils. Histamine also promotes vasodilation of local arterioles to increase blood flow to the injured site and permeability of the local capillaries. All inflammatory chemicals dilate the local arterioles and make the capillaries leakier. Many also attract the various phagocytes to the area, and some will mobilize lymphocytes and/or trigger the adaptive immune system.
Okay, I couldn't keep up with that, but I'm trying. So
Sorry, I can read slower.
That's all right. No, that's okay. I'm listening for the key things and capillary leak and vasodilate, arterioles. Okay, so let's go back to the various ways that you can trigger initial inflammation. So this is before you get anybody there. So you mentioned basophils, but they're not there yet. Mast
Right.
cells.
I started with just the chemical alarm from the damaged cells.
Yes. So let's say you cut your skin, like she was talking about, and you're going to damage some of the membranes of both, probably even the dead cells as well as the living cells. And that's going to cause prostaglandins, and you're going to know that because it's going to trigger pain. You just cut yourself. You may also damage sensory endings, or, the tactile sensory cells, and that'll give you a pain response. But in addition, it releases those echinocanoids, the fatty acids that Dina mentioned. And those are prostaglandins that act to call in white blood cells and trigger inflammation. Mast cells, now when mast cells, if they're not damaged themselves, what causes mast cells to start to release their histamine that we talked about? Anyone, Dina, or anyone else? They have to bind something. Acronym.
The PRR to PAM.
That's right. When their PRR binds a PAM. Now, they probably also respond to cytokines, but the book doesn't bring that up, so I wouldn't ask you. And then pathogens themselves, and Dina was great at bringing this up. Some pathogens release enzymes because they're going to eat you, right? And those will release the prostaglandins, or even certain chemicals are released by, there may be waste products from the pathogen's metabolism, which are pro-inflammatory. So the pathogen itself can trigger inflammation. Was there anything I missed? As generally, we have 3 ways here, that because I know I wasn't able to keep up. Let
me see. Let me see, what do you have? You've got the lysozymes, the body secretions, present in body secretions, as well as neutrophils and macrophages, are the defensins and the lysozymes that act to disrupt, that act against cellular pathogens by disrupting and damaging their cell membranes and inhibiting bacterial and fungal growth.
Yeah, no, that's all true. I don't know that they actually cause inflammation. I'm not sure if any of the membranes of a pathogen would have echinocanoids that act, but I don't know that. It's possible. And so I'm going to leave it off because we really know at this point. They already have enough, but it was good information. Okay, so then she did a good job of bringing up what capillaries are getting leakier. I probably should have done this a different way. Let me erase some of this. But you got to vasodilate first. And why do you want to vasodilate? Let me just go back to one being you vasodilate the arterioles. So that's going to bring more blood in there, right? Increase blood to area. So what is in the blood that you want to have arrive there?
The white blood cells.
Yeah, you want your white blood cells. And you want plasma, because in the plasma, as you'll see if you look at the table, there's a lot of both antimicrobial and healing, and you're also bringing oxygen, you're bringing red blood cells, right? And nutrients, because you're going to fight a war, you've got to have the supply train increase. And that, of course, means you're sending more into that area. So that's great, but you do want those white blood cells. Now, the white blood cells in the plasma can't leave unless the capillaries start to get leakier. That's especially true if it's a continuous, which most of your capillaries are, right? So they have lots of tight junctions. So to get leakier, you have to decrease the tight junctions. And why do you want to do this is you want the plasma to bulk flow more readily out, but you also want to make space for the white blood cells to skinny their way between the endothelia. But it still will prevent albumin and red blood cells from escaping through those. It's not like it becomes sinusoidal. It's still holding back large things. You do one other thing on here to the capillaries, and maybe you said it and I missed it. What happens to their endothelia? Did you say it, or should I just go?
I don't think I said it. I mean, it just, the permeability of the local capillaries increases with the vasodilation.
Okay, and they spout sites on their membrane for the white blood cells to stick. So I'm just going to say get stickier. And what do they do it with these cell adhesion molecules? Those are called CAMs, if you might remember from 103A. So in order to leave, the white blood cells, remember, it's like they have to get an off-ramp. The blood is flowing through them, granted not super fast because this is a capillary, but still, you saw the video, it's moving along there. So those white blood cells, whether they're neutrophils or eosinophils or monocytes, they have to be able to stop and get off of the road of the flow of the blood. So the endothelia, when it detects inflammation, pro-inflammatory chemicals, it starts to spout these membrane proteins called CAMs that allow the white blood cells to stick. And I think the next group is going to be eventually talking about this process called diapedesis and the exiting. I think it's the group D. So the last question here, 8, is what white blood cells and plasma proteins themselves increase the inflammation? Now, we started the inflammation back here, but now who are going to increase enhancing inflammation?
Well, I've got after the plasma components and white blood cells arrive, kinins, which is a plasma protein, urine. It's found in the plasma, urine, saliva, and lysosomes of neutrophils. And they also increase vasodilation of the local arterioles to increase blood flow to the injured site and permeability of the local capillaries. Kinins also induce chemotaxis of leukocytes to attract them to the injury and induce neutrophils to release their lysosomal enzymes in a positive feedback that increases the production of kinins. Kinins also create or increase the pain sensations. And once the inflammation response is created, the complement system is triggered. And then I've got the mast cells release histamine to promote vasodilation and increase blood flow and formation of edema. Basophils also release histamine. Neutrophils also produce or release histamine. The neutrophils also
Book? As far as I know,
I think I found that in the book.
It was in the book?
I think. I'm not sure where I found that, to be honest. The neutrophils, I think the most important thing is the kinins. And then all cell membranes and generated enzymes of neutrophils, basophils, et cetera, produce prostaglandins or release prostaglandins. And then the leukotrienes by the neutrophils and macrophages. And then the lymphocytes, I have TNF-A alpha is released, and then the liver C3A.
Are those inflammatory?
C3A enhances inflammation by inducing more histamine release from the mast cells and basophils.
So if you look at your table, C3A is a complement, so maybe I
Right.
shouldn't put it under there. This is a complement. We'll come back to that one later. So we'll come into that one, as I said, a bit later. So what else did I miss here? Complement.
Well, we talked a lot. I mentioned prostaglandins. We talked about that a lot in the previous
Yeah.
section. The kinins we talked about, the kininogens.
Necrosis factor.
Kinins I've got, they're converted by the plasma protein kininogen, which is produced in the liver, and that promotes vasodilation, leak your capillaries to increase blood flow and formation of exudate or edema, and then induces leukocyte chemotaxis to the area.
Okay, great. So I think that's pretty good let's see, this is a white blood cell. This is plasma. This is in the plasma. And this is eventually this is from the macrophage, right? Yeah, from the macrophage. So that's a pretty good list already. So I'm hoping that what you're getting from this is one, that there's lots and lots of ways to increase inflammation. And all of the ways down here are from your innate response. So this is like a snowballing effect. Once you start inflammation, you have fluids and cells leave your blood into the tissues, and that promotes more inflammation. So part of that's because you're going to increase the number of neutrophils that damage more cells. So then those initial damaged cells are still there, but now you're getting more damaged cells, including the dying neutrophils themselves. And they release more prostaglandins, like what initially started it. Now, basophils are in really low numbers in your plasma or in your blood. Remember, they're less than 1% of the white blood cells. However, if the inflammation is more chronic, so it goes on long enough that your colony stimulating factors can tell the bone marrow make more basophils and it manages to make them, then you're going to see basophils really having a bigger effect. And that's why you'll see in the differential white blood cell counting that things like parasitic worm infections, which tend to be a chronic infection, and allergies, which, can go on for a whole season, depending on what you're allergic to, those can tend to increase basophil numbers because the inflammation response goes on long enough that the bone marrow starts to make a significantly increased number of basophils. Now, these guys, as she mentioned, but I probably forgot to write, they also release histamine. So they're like a mast cell that's coming out of your blood. I didn't put mast cells because they're already there and they're already being triggered. I suppose there more white blood cells may trigger more of them. So she's right in what she said, but since it was already in the earlier list, I didn't put it back again. Kininogens are made by the liver if you look at your table. So the liver makes these. And once again, hopefully you're starting to see the theme. If you see an O-gen at the end of the word, these are in an inactive form in the plasma. That means that white blood cells have to activate them. And I think it's mostly neutrophils. Did you say that, Dina, when you talked about the kininogen?
Yes.
Okay, so the white blood cells that have left and are busy releasing various granules, including these neutrophils, will release an enzyme that will cause the kininogens to become kinin. So kinin is the active form. And she really did a nice, thorough list of all the things that these kinins can do. The book specifically brings up bradykinins, which if you want to learn, you can. I don't ask you to learn a specific kinin in this case. So just know there's a bunch of them, and they have lots of actions, including increasing inflammation, which we're focused on here. But as she said, they can encourage the complement that we'll talk about, and they can cause the chemotactic gradients, all the things that Dina kind of went through. I tend to focus in your table on their inflammatory, pro-inflammatory effects. Okay, so we will be coming back to complement and C3A. Macrophages, once they're activated, so these free macrophages that come in, they start to release tumor necrosis factor alpha. There are multiple tumor necrosis factors, so the only one on your table is alpha. And if you look at its action on the table, it says promotes inflammation. So it does. It's also a hallmark of inflammation. If it's being released and you're a diagnostician and you see higher levels of tumor necrosis factor alpha, that may be because of inflammation being a kind of chronic inflammation event occurring. You can see that this is sort of a positive feedback, although I don't ask it in that terms, in the sense that once the macrophage releases the tumor necrosis factor, it tends to increase the activity of many types of white blood cells. So in addition to causing or promoting inflammation, it may be doing that indirectly by causing neutrophils to be more activated and basophils to be more activated and mast cells to be more activated, and therefore it may be an indirect. So we don't go into that much detail. I'll put a question mark there. Okay, so that was great. You did a great job on that. And I don't want to undercut inflammation in any way, even though we're going to move on from inflammation here to back to phagocytosis, because clearly this must be a really important response for you to get a good innate defense going, because it's bringing in your white blood cells and your plasma components that we're going to talk about in a minute. However, you also know that it's not a fun thing to have. So people tend to take a lot of inflammation suppressing drugs when you have an inflammatory event. Now, sometimes that really is helpful. Let's say you twist your ankle and there's no pathogen involved. You still damaged tissue. So you start an inflammation event and you need to bring white blood cells in to clean up the mess of the damaged tissue. But you may also know that chronic inflammation can lead to more scar tissue being deposited and can lead to calcification in places that you don't want calcification, like at the ends of bones, and you get bone projections. So it's a good and a bad thing. So sometimes you want to suppress inflammation for comfort, or even to prevent scar tissue formation in areas you don't want scar tissues to form if you have some kind of chronic inflammation. Okay, any questions on this? Let me get chat going again. It disappeared somehow, because I have to get to the three little dots wherever they are. There they are. Let me see if I see any chats. Or just speak up if I'm too slow at finding my... All right, so let's move on to group D, and we'll probably just get through, hopefully, D and E, but we'll see. So group D, do you have a spokesperson?
Yeah, Professor, I'll be doing question 9.
Okay.
Which, question 9 is asking us to describe the process of diapedesis and name several cytokines that enable the chemotaxis that occurs. So diapedesis is a process that happens when phagocytes are mobilized. And this is when phagocytes flood the area of injury based on the inflammatory chemicals that have been released in response to injury. So leading up to diapedesis, we first have leukocytosis. And this is when the injured cells in the area release leukocytosis-inducing factors. Sorry about the background noise.
Yeah, you might have to speak a little louder.
So the injured cells in the area release leukocytosis-inducing factors. This causes the number of neutrophils to increase by 4 to 5x. And
Okay.
then next we have margination. And this is when the phagocytes cling to the sides, the inside walls of the capillaries, which cause the endothelial cells to sprout those adhesion molecules, causing the neutrophils to stay in place. And then those neutrophils that are in place then get activated by inflammatory chemicals, and they start releasing more adhesion molecules. And then diapedesis is what occurs next. Because the inflammatory chemicals are present, this signals the neutrophils to flatten and squeeze through the
I'm spelling that wrong. Right?
So the neutrophils flatten and squeeze through the endothelia of the capillary walls, and that's the actual process of diapedesis. And this occurs due to the chemical gradient. So that's the chemical gradient that exists of the inflammatory chemicals cause the neutrophils and white blood cells to move out of the endothelia of the capillary walls and towards the site of injury and start phagocytizing.
Okay, that's brilliant. So there is a nice picture in the book, and I think I'm spelling diapedesis wrong, but just double check me on that. So we talked about these earlier ones, and Sana did a great job of summarizing, that you need to have some sort of gradient. This is only a couple examples. There was all sorts of things mentioned, including the kinines and interleukins that can cause this chemotactic gradient to be set up, and also, besides triggering inflammation, can help signal white blood cells as they pass by that they need to leave the blood and enter into the tissues. Now, in order to do that, they have to glom on to the endothelial cells. So the endothelial cells change, sprouting CAMs, and the more the white blood cells bind, like Sana said, the more CAMs appear, and they then can get off the highway of the blood, and eventually start to flatten themselves out and crawl between the endothelial wall, which is what the diapedesis is really describing. Once they're outside of the capillary and the bloodstream, they can follow this chemotactic gradient in order to get to the area where the damage has occurred and therefore is causing this gradient. Okay, are you still doing ways of increasing healing using inflammation? So
But let me just quickly, I think I didn't talk about the specific cytokines that enable chemotaxis. So that would be.
Right, you were supposed to do that too. Let me write that down. Okay. Chemotaxis via what were.
So that would be via prostaglandins, leukotrienes, histamine, and then the TNF alpha.
Okay, and the kinase too. That,
Oh, right.
that Dina mentioned. Okay, there's great. I think it only asked for 4. Well, name several. It doesn't say all of them. So check your table, and you can always add to that. Okay, so how about this healing? What are you using inflammation as far as allowing for healing? What does it do? Okay, great. Great. Okay, all that fluid coming out is going to cause a local edema or swelling, as she mentioned. It's going to bring antimicrobial things from the plasma. And the white blood cells, these are the white blood cells that come in and are phagocytic, especially the macrophages, will eat up not just the pathogens, of course, which neutrophils will do, but there's really actually, which the book doesn't talk about, two types of macrophages. One specializes more in going after the pathogens, and another one more at finding dead and pieces of dying cells and disposing of our own killed. They're kind of the, I'm blanking out, not the medical examiner, but the person who comes to take the bodies away after a death occurs. So there are certain types of macrophages that specialize in not looking for PAMPs and with PRRs, but rather in getting rid of the debris. And the last is that it brings in the nutrients and growth factors. Remember the platelets releasing growth factors that help for healing. So not only are the nutrients supplying energy and oxygen to the active white blood cells, but also to the tissues needed for healing. Okay, so let's see the time. We have time to start complement. Hopefully, we might have to supplement this, but this is Group E. So who is the spokesperson for Group E? Do you have one? You can go back and find out who Group E was, I think, if I can get this thing to work. Somebody in Group E? I don't have time to fool around here.
I'm not in Group E, but I can go ahead and answer 11.
Okay, I'll find out who the cowards were in Group E here. Okay, so how about the 3 ways that you can cause C3 to form? Maybe Noah, tell me about what C3 is too.
Well, C3 is like one of the plasma proteins, like complement, and it's triggered by 3.
Not exactly. It doesn't exist in the plasma. It has to be formed, right?
Right.
By these pathways. So you think of complement almost like a bunch of, because I had a son, like a lot of little Lego pieces.
Yeah, I remember you saying that.
You got to put together. So you're putting things together, and we don't have to know the numbers of them, but you're going to have many subunits.
Right, and they're made into liver. Some of them are, at least.
And what did you just say?
Some of those subunits are made into liver, like lectin.
Right. The liver is making these subunits, and it's in the plasma, inactive in the plasma. Okay, so now we'll go into, it's circulating the plasma leaks out, and there are 3 pathways that you can make this active form. Well, it's sort of almost active, I suppose, but you have to make this stuff. So go ahead, Noah, what were you
So the three pathways are the classical pathway, the lectin pathway, and the alternate pathway. The classical pathway gets activated by antibodies coding a target cell. So it's like an antigen antibody complex.
Mhm. So for classical, you need antigen antibody binding.
And if I recall, like it doesn't matter if it's cellular or acellular.
Mhm. Plus subunits. And you end up with C3. Okay, and that's good that you added that. The antigen can be on the cell or it could be something released by the cell. Doesn't really matter whether it's they call it a soluble antigen, meaning it's something released. Maybe it's a waste product or a toxin released by the pathogen, or it's actually physically bound to the pathogen, as long as the antibody will bind. Now, who the antibodies are will come back when we do the adaptive. So this is critical, and you end up with C3. You need some of those subunits. Again, we don't need to know which ones. They are given in the book, but we don't need to know which ones. Okay, what's the next one you want?
One more thing I wanted to add it for classical is that it performs best in like secondary infections or late in the primary infection.
So that's great, adding that fast in secondary, slow and not as strong in the primary. Again, because it requires the adaptive. So that's going to be a theme. If you have a secondary response, you need antibodies, then you're going to be a lot faster and stronger in secondary. Okay, what's the next one that we...
So the second one is the lectin pathway, which is activated by lectins binding to specific sugars on the microorganism surface. Specifically, a common sugar would be mannose. And this one does require a cellular organism. Lectin is also produced by the liver, and it's activated by macrophages.
And then you need plus subunits, and you're going to get C3 again. Okay. All right, this guy, lectin, as he said, is made by the liver. The liver is told to make the lectin by the macrophage. So it's unlike the subunits, it's not always making lectin. It's triggered to make lectin, signals maybe, to release that lectin. So you do need the macrophages being activated before they're going to do that. So I don't know for a fact which signaling molecule, whether it's some type of interleukin, or at one point the book had said the tumor necrosis factor alpha, but I don't see that anymore in our book. So an activated macrophage, it's already phagocytic, it's out in the tissues. It signals the liver, make lectins. Lectins enter the plasma. The plasma leaks out in the inflammation response, and there's a very common sugar. Now, they used to always just say mannose because it used to be called the lectin-mannose pathway, but I guess other sugars can do it, but not a lot of other sugars. So mannose is fairly common sugar that might be in, if it has just a lipid bilayer on the outside, might be present there, or even in the capsule on the outside of a pathogen. And so this is a fairly non-specific to any particular type of pathogen, but as he said, it has to be on the surface. It can't be, unlike the classical where you can have a soluble antigen and trigger it. Here you have to have something on the physical surface of the pathogen. I know we have one more pathway. Can we just quickly do that one so you'll have well into that? I know we'll go a little late, but
sure,
the last one.
It's the alternate pathway, and it's activated spontaneously because there's a lack of concentration in inhibitors from microorganisms. Because it's pretty much like on a hair wire trigger. Once there's a lack of inhibitors, it pretty much just goes off.
Okay. So our own cells suppress this pathway. So it's suppressed by our own cells, suppressed by healthy cells. I'll just say by healthy cells. But when you have pathogens present and they don't have inhibitors on the outside, you can get this. Now, when I taught micro, what I used to hear was the inhibitors are usually on the cell surface again. So this isn't something secreted, so it's on the pathogen. And the typical pathogens that have inhibitors have cell walls. So this is the way at least I learned it. Kind of limited to organisms that have cell walls, so fungi and bacteria that have cell walls. So something like a virus wouldn't be able to activate alternative pathway, and it wouldn't be able to get the lectin pathway, but classical pathway could do it. So you can see that all this again needs subunits, and then it results in C3 again. So 3 different ways of getting C3, and they're kind of situational. If you had a viral infection, you'd probably use the classical pathway. If you have a cell wall or some surface, it doesn't have to be a cell wall, but some surface that has mannose and you're a cellular infector like a bacteria or a fungus, not a virus, then you could do the lectin mannose pathway. Similar with the alternative. Now, the alternative is the least specific. It's not looking for any particular antigen, like this one needs the antigen. This one needs mannose to be there. This one doesn't need anything to be there. It just needs a lack of something. And why would it have an inhibitor? Well, these microorganisms are evolving alongside us, and so they try to suppress C3 activation, and therefore complement activation. So complement is a really powerful part of your innate response, and it's very effective. So when we come back, this group, and maybe I'll go look it up before I leave so I can prod somebody who was actually in group E to tell us about the actions of C3, A and B, and that will finish up the first essay. So I think you have plenty to work on for the first essay. The hardest part on complement is this part that Noah did for you so well. So why don't I meet you back for lab? Remember, you're drawing slides. I'll show you some of the photographs of the one model we have and talk about it. But other than that, we're going to be drawing slides. The lab manual doesn't have, it used to, but it doesn't have good pictures anymore of these slides, but the textbook does. So you might want to have your textbook out so you can see some of the things that need labeling and where they are on the slides. Okay, I will see you in about 10 minutes if you're in that, want to come to the first lab, and about 1 o'clock for the rest of you. Oh, I see what I did wrong. So I don't see anything. Can you repeat what I said? Words ending. Oh, ogen. So a while back, someone asked about things ending in ogen. Almost always these are released by the liver. And when they add the word ogen at the end, like fibrinogen, angiotensinogen, you're talking about an inactive form in the plasma that needs to be activated by some condition change, like renin for angiotensinogen. Okay, again, I'll see you about 10 minutes from now, and we can